Hi, hello everyone. So welcome to our presentation. I am Dr. Adrien Oliva from CSIRO, and today we'll be talking about APIs and their power in genomic research and how they transform the field of genomics. So like I said, I'm part of CSIRO, but most particularly, I'm part of the eHealth team, which is the Australian biggest digital health program. We have a wide range of products and services. Some of them were presented at previous Apidays, actually. And the main services we have are OntoServer, which is the world-leading clinical terminology that is used in more than 80 countries in the world. And also CardioHub, which is the first online cardiac rehabilitation platform to support cardiac patients in recovery. And our goal as a team is to improve medical diagnostics and medical treatment or everyday life as a whole, all of that using genomic data. But if I want to explain how APIs impact genomic research, I have to quickly start by introducing what's genomic. I'm going to try to make it quick because I know biology might not be funny for everyone here. So to start, what's genomic? Genomic is it's a study of DNA, right? And DNA can be seen as the blueprint of life of any living organism, as it has every information and every things that makes those organisms being alive, right? And you should try to see your DNA as your own storybook, and the most important storybook ever written about you, as it contains information about your past, your present, and your future, but not about just yourself, but about your entire bloodline, right? So as you can guess, these data are very sensitive and they should be protected at any cost. Because if the data finishes in the wrong hands, this can become dramatic, as information such as ethnicity or physical trait and even predisposition to diseases can be extracted from it, right? But in the good hands, scientists and researchers are able to study the DNA to get insight and to understand the disease, develop more effective treatment, or even predict and prevent some diseases. But the discovery we make is directly correlated with the amount of data available. And with the cost of sequencing getting lower and lower every year, we are having more data than ever to study. And that's why we are getting better and quicker responses. And the best example is the COVID-19 outbreak, where genomic played a critical role at stopping the spread and developing a vaccine at an unprecedented speed. But the amount of data I'm talking about today and about genomic is huge. And to give you a rough idea of how much data I'm talking about, roughly, genomic every year generate 20 exabytes of data. And to compare, as a comparison, YouTube estimated database since its opening in 2005, it's 2 exabytes. So on average, genomic create 10 times more data per year than YouTube ever did since it's been created, right? And most of those data need to be protected and analyzed, and this can be challenging, and it is challenging for researchers. And that's where APIs and cloud solution comes in. And so today, I'm going to talk and to present you 3 different applications from our group that showcase how we use APIs and cloud solution to drive impact in health and genomic data security. And I'm going to start by talking how we use APIs and cloud solution in general, right, to connect the data to the analytic tools in order to advance research and clinical genomics. So to do that, let me introduce to you ViSAP, which is a machine learning framework for genomic data. And this software is used to find genetic association with a trait or a disease. In other words, it's trying to find mutation in the DNA correlated with eye color, BMI, cancer, or any kind of diseases. So how does it do that? First, you need 2 sets and 2 cohorts of people, one with the trait that you're analyzing and one without. Then, using that, ViSAP use machine learning algorithm and random forest, to be more precise, to find part of the DNA where the 2 groups are different a large number of times. And doing that, we're gonna do some statistical analysis to find if those mutations are meaningful. And if they are, we just find a correlation between the DNA mutation and the trait that you're analyzing. But where our software is different from other available is that we can run on top of that ViSAP. And what ViSAP is doing is trying to find interaction between genes. Because, for example, today we know that your eye color and my eye color and every stuff in every genes in general is not working alone. And eye color is controlled by 6 different genes, and 6 different genes are working together to decide your eye color. And that's pretty much what's happening in the body and in the human everywhere. And that's what ViSAP is trying to do, is trying to find interaction between genes associated with a trait. And once it does that, and we can do that for 4 SNPs, 4 different genes at the same time, we can correlate those interactions to a disease. And this can sound kind of easy to do, but to give you an idea, a human have 3 billion DNA in the letters in their DNA. So when you're working with hundreds of thousands of people, this amount of data can become impracticable very quickly. And that's why VariantSpark is mainly run on the cloud. And this is the basic architecture of VariantSpark on AWS, but it's available on all major cloud providers. And here, in this architecture, APIs allows us to access the large dataset safely and allows us to send it to multiple instances to execute VariantSparks in parallel and run it quicker than other software. Because, like I mentioned, we are not the only software to run this kind of analysis. Those analyses are called GWAS, Genome Wide Association Study. But we're not the only one, but we are the quickest, as you can see, as yellow and blue are VariantSpark. But most importantly, we are scaling linearly, where other software are scaling exponentially. And this difference in scalability can be the difference between being able to run an analysis or not, which can be the difference between being able or not to find insight about a disease, right? And all of that is possible thanks to the architecture and APIs and cloud solution, allowing us to access and store those data secure in a safe way. And now, as you might start to understand, as researchers, we like having data, lots of it, as it means more discovery and more insight. But data, especially in genetics, always come with consent. And this is mandatory and also understandable when you understand the reason I explained at the beginning of the talk. So it's primordial for us as researchers to create a robust data protection to safely guard sensitive genomic data, but also to create trust between researcher and patient. And now I'm going to present you a protocol that we developed with my team to do just that. But first, I want to explain to you what's happening today when we go to an hospital or a clinic, right? So if I enter to an hospital today, they're gonna come and they're gonna create a file about myself, and they're gonna get a blood sample or a DNA, and then they will send this DNA to another lab. And this lab will do the analysis, but they will keep a copy of this DNA. This lab can also send it to another sequencing facility who will keep a copy of my DNA too. And when I go to another clinic or another hospital, this will happen again, right? And then you will have multiple copies of your DNA. And at this point, you would lose a complete data ownership over your DNA, and you don't even know where your DNA is at this point. So that's what's happening. But what can also happen today is that if you are part of a cohort, one central organization will control all your DNA and all the DNA of the cohort, right? And this is also an issue as it raises security concern, as it gives too much power of the organization. And with the rise of blockchain and decentralized technology today, we don't really like having sensitive data in a centralized organization. So with my team, we try to develop a pipeline that first and foremost has to be secure, which means that the patient can access and store the data safely. We also want the patient to give the possibility to the patient, to give consent, to remove it or to change it at any time. We want the pipeline to be transparent, which means that anyone can know at any time anything that they are allowed to know. The pipeline has to be auditable, and it has to keep track of all changes that happens. But most and foremost, we want to give data sovereignty to the patient and create a patient-centric pipeline. And in our case, we got the chance to work on a project involving newborn screening, where new parents have a choice to sequence or not their kids' DNA for screening purposes and check if the newborn have some diseases and all. And then from there, we will ask them if they want to enroll into our protocol and to share their data to research. And our protocol is called Dynamic Consent Protocol. And based on voluntary, obviously, if the patients agreed and enrolled to our protocol, they will have access to their interface, where they will have access to educational videos and a chatbot where they will be able to discuss. This chatbot was developed by CSIRO e-health team, and it was specially developed for this matter. Patient will also have access to this web page, which is the page where they can give the consent. And our goal on this web page was to be able to give granular consent, which means, for example, the patient can share the data for cancer research, but if they want, for example, they can not share the data for ethnicity and population genetics research, right? But most importantly, from this single page, they can revoke every access to anyone they want, but they can also remove their data from this single page from anywhere. And that's a big change. And this change of consent and this consent in general is stored on a blockchain technology to make sure we keep track of all changes. And the patient also have access to a dashboard when he will see the preliminary result that his data allows a researcher to find. And to find and to access this interface, data will use API calls through a mobile app called MacroKey. So this mobile app is developed by Data61 team from CSIRO. It's a self-sovereign identity app, and you should see that as a digital wallet that stores unique credentials for the system. And using this app, the patient will be allowed to access the consent and the dashboard and all of his interface. But you won't be allowed to access other data, as the other data from other patients will be stored on the cloud in a different location. And that's where the data processing and the screening and all the analysis will take place on the cloud within the institution and the organization. And to access those data, a researcher, clinician, and doctors will access it using the same app, MacroKey, and using an API call once again. But they will access the data using an engine, a new engine called SBeacon. And this engine allows us to query the data, but not access it, which is very different, and I'll explain that later. And that makes sure that researcher and clinician doesn't do what they're not supposed to do with the data. But finally, they will be allowed to have an ongoing discussion with the patient, not directly, but they will be able to access the dashboard to update the result and the preliminary results that the data allow them to find. All right, and that's how you create a transparent and you give back the power to the patient over their own data. But now that we have data and that we have the consent from the patient, we need a safe and a secure way to share and access those large and diverse datasets while keeping the data secure. And that's where Beacon protocol comes in. So Beacon protocol was created by GA4GH, and the idea was simple, right? For research to move forward in the direction for better personalized medicine and more effective treatment, we need to study and access those sensitive data, right? However, we can't just lock this data off just to keep it safe, right? If we do so, research will never move forward and science will never progress. So that's why Beacon API is here. Beacon API is there to facilitate the search for genomic variant and metadata without compromising the privacy of the dataset. So how does that work? Well, most of the time, a researcher will have a specific question about DNA. And this tool, a researcher will be able to ask this tool, for example, where was it a mutation at position 10 at the chromosome 5, for example. And the tool and Beacon will give us back, yes, there is a mutation, or no, there is no mutation, obviously. And it will give us information such as 10 individuals out of 1,000 have the mutation, and they will give us metadata information, such as 2 of them are sick and 3 of them are male, and so on and so forth. So it will give you information about the group, but never individual information, and that's how we keep the privacy intact, and that allows researcher and clinician to do the research without giving and raising any security concern. And our team created the serverless version of this beacon protocol called sBeacon. By doing so, and by creating a serverless version of it, we allow researchers from all around the world to exchange genomic data safely and efficiently, allowing us to transfer information between different institutions and organizations. But one of the main features is the ability to share this data while maintaining the complete control within your own infrastructure, which can be a crucial consideration for many organizations in the field. And by making it serverless and on the cloud, right, sBeacon can be scaled up and deployed for just a few individuals, just 1 or 2 or 3, or like a few individuals, to a large cohort of millions of people. And like I said earlier, one individual has 3 billion positions, right? And we were able to scale up to 40 million individuals. So we can integrate 40 million individuals to 3 billion locations. But what makes it good on serverless, to be a serverless beacon, is to have a quick answer, and every query takes less than 1 second to update. And also, Chip we don't have idle cost, and for huge data sets such as the 40 million one, if you do that on premise, the idle cost will be very expensive. And there is multiple implementation of Beacon in different organizations, and this is a comparison of their query capability. And on top of sBeacon being able to scale up more than the other, what set sBeacon apart, its adaptability with onto server, which I introduced earlier, and perhaps even more importantly, it's its compatibility with dynamic consent protocol that I discussed just before that. And this protocol allows patient or institution to share the data and to change access to this data based on their consent on all or just a part of the data in real time, which is a big change and a big deal in the field. But if you have more questions, or if you are just curious about the project, sBeacon has been officially published in Nature Biotech last month as the first production-ready implementation for large-scale genomic data exchange. So feel free to dive into the paper or to reach out to me if you have any question. And to finish and to wrap things up, I want to summarize the key takeaway from my talk, and 3 of them. But APIs is involved in the field in more than 3 ways. But generally, APIs in cloud solutions have revolutionized the world of genomics research by affording us the ability to analyze and access an unprecedented wealth of data, which lead us to the identification of crucial diseases-related genes, allowing us to find better treatment about diseases, right? But they also enable everyone to access data securely, and that's placing the data control back in the hand of the patient and granting them data sovereignty. And lastly, the secure sharing and access to data through API and cloud solution created a collaborative workplace that drives innovation and progress in the field of genomics. And that's how APIs revolutionize the world of genomics in general. So I want to thank my team that work on the different projects that I presented you today. I want to thank all the organizers of Apidays 23 for giving me the chance to work today and to talk about our different projects that we develop in my team. And if you want to reach out to us, or if you have any question, you can reach us to my email just there, or to the Twitter or X. I don't know how to talk about, or to say it today here. And yeah, happy to work with you.